Yeah, so hello everyone, my name is Mashbayar, and today I'm going to be presenting our Connectome to Model tool, which can take any arbitrarily connected directed graph and turn it into an artificial neural network in 2 lines of Python code. So before we get into the details of our model, let's talk a little bit about our motivations behind it. So, as we all know, AI has been developing very rapidly over the recent years, and ANNs are nowadays quite proficient at tasks in many different domains, from image recognition to speech recognition and speech prediction and so forth. But these ANNs are often quite dissimilar to the brain, particularly with respect to its linear, often feed-forward only architecture. And possibly due to this dissimilarity, you often see ANNs struggle with more naturalistic tasks, with more challenging and obtuse stimuli, such as occluded and cluttered stimuli, so to speak. So in work project 6, we wanted to test whether or not human brain-derived macroscopic architectures gave any advantage to ANNs for human-relevant tasks. So in order to test that, we want to be able to iterate. We need to be able to make quickly make ANNs from multiple different graphs. So that was why we made our Connectome to Model tool, which can turn a directed graph into an ANN, and also particularly an ANN that in each layer has 3 different biologically based inductive biases as well. And these inductive biases are local recurrence, topography, and modulatory top-down feedback, each 3 of which we'll go into. So first of these inductive biases, we implement the local recurrence we implement by modeling each area or each node of the connectome using a gated recurrent unit, which is a standard locally recurrent architecture in AI. And it is able, with its hidden memory and its local recurrence, it is able to capture the temporal features of the stimuli. However, the normal gated recurrent unit usually does not capture the spatial features of the stimuli. It doesn't have any kind of topographical features, so to speak. As such, we use the convolutional version of the gated recurrent unit, which can capture the spatial features of our stimuli as well as the temporal features. And our final modification to the gated recurrent unit is its input. Not only does the conv group receive the traditional feed-forward input, it also receives a modulatory feedback input, which does not drive the activity of the cell, but instead element-wise multiplies the activity that is driven by the feed-forward input. And it's a distinction that is derived from biophysiological studies in layer 5 cortical pyramidal neurons. So this is the kind of ANN that you're going to have. That's going to be the result of our tool. But how do you make? What is the process of making that ANN? So for that, I'll give a little bit of a sample workflow. So first step, you want to determine which areas of the brain that you want to model, and particularly figure out the connections between them. Using, for instance, structural data, diffusion-weighted imaging, so to speak. And after you have the connections between area, you want to figure out the directionality of these connections. Who feeds forward to who, and who feeds back to who. And in our case, because we're working with humans, we did it using qualities such as cortical thickness and laminar profile, qualities which are known to follow a sensory associational gradient. And we use these features to sort of hierarchically rank our areas. And now, once you have this directed connectome, then you're going to be able to use it in our tool to build your neural network. So that's all fine and dandy, but what can we use this ANN for? What kind of questions can we answer with it? So a little bit of an example of that. I don't know about you guys, but I have a really bad vision, and if I don't have my glasses, then sorry, you're all like blurry, ambiguous images. Unless, for instance, you speak to me, in which case I can recognize you by your voice. And somewhat inspired by that, we designed this multimodal task where the model receives an auditory stimulus and a visual stimulus, and is asked to identify what the visual stimulus is. And sometimes the visual stimulus is ambiguous and blurry. This could be a 3, this could be a 5, this could be an 8. But in that case, you will always have an informative auditory clue that can give you the answer to what this visual clue might be. However, crucially, there are also cases where you can just use the visual input by itself. It's clear enough. You don't need the audio. And in fact, the audio might be completely wrong. It might be distracting, in which case you need to be also able to ignore the audio as well. And very important to note is that at any point during the training and testing, the model might be receiving either of these scenarios. They are not told which of the scenarios they're facing. They're just given the inputs. We trained multiple different models with this tasks. We've trained 3 different randomly connected models and a brain-like model, where and a particular feature of the brain-like model, which is due to the very distinct laminar profile and average cortical thickness between the auditory and the visual regions, is this clustered kind of graph, so to speak, where the auditory regions only feed back to the visual regions. So there's no such thing as the V1 feeding forward to the A1 and then the A1 feeding like forward again to the visual area. So there's this clustered connectivity in the brain-like graph. But otherwise, all the parameter counts and the connection density is all the same. All the per layer parameters and the details are the same as well between all these graphs. So is there any difference in performance between any of these graphs on our multimodal tasks? So interestingly, in the case where you have to identify the ambiguous image using the audio, pretty much everyone learns it, and fairly consistently at that. However, in the case where you'd have to ignore the irrelevant input, then the brain-like graph is able to learn this much more quickly, and also much more consistently across 10 different seeds compared to the random graph, where their performance very heavily depends on what weights they're initialized with, and varies quite considerably. And it suggests that this biological clustered kind of connectivity, where the connection mediated by feedback allows the visual and auditory, these different modalities, to influence each other but not override each other's stimuli. So that was an interesting kind of thing to find out with our tool, but that's only a little bit of a tip of the iceberg. So we're interested in exploring a lot more different questions. For instance, what if you compared a human connectome versus a macaque connectome on a human relevant task? Would you see a difference? What would additional brain-like details do? What would different kind of top-down mechanisms do as well? And also, probably most immediately for our group, what would brain-like graphs do in a sensory motor task that is reward-based, as opposed to the more AI-like supervised learning regime that we used for this task? And these are the questions that we're interested in, and I hope that you guys will also be interested in asking your own questions using our model and ANNs as well. So thank you very much, everyone, and questions?
Any questions? Actually, oh yeah, go ahead.
Nice talk, thank you. How do you validate, any ANN architecture produced by your models? Because it's one thing to show that on a particular task they perform better than a random network, but how can you say that, you know, it says something about the brain at all?
I'm wondering, by validate, do you mean validate the performance of the test, or validate its similarity to the brain?
Both. I mean,
I see.
If I had a generator of some model, some machine learning model that was completely different from yours, how could you say that your generator is better than mine?
Well, one method is just like kind of how you do in neuroscience to run like different trials, particularly using like different initializations and potentially different parameters, so to speak. So in our case, in the test that I showed earlier, our method of like validation was, trying it with different initializations, because potentially, you know, one mouse just could be better at the task than the others, and one set of initializations could be also better at the test than the other. So we want to test it on these different kind of seeds, so to speak, right? And with your second question, kind of like saying like saying something about the brain. Yeah, it's always a question in your AI like how much of your details to include in your model, and how much at what point can you say that your model is really brain-like? Like, in my opinion, like the approach that I take, which Blake also mentioned earlier, you wanna in my case, in this case, we held everything similar except for this kind of clustered connectivity, right? Except for this part where you only receive feedback from the auditory. In that case, you can sort of isolate that as the cause of why there might be difference between there might be any difference between that model and the other random models, and so you can get a kind of a explanation out of it, an explanation of a biological phenomenon. But yeah, that is a very good question. It's always a question in neuro AI whether or not how much of the brain to include, and how. Yeah.
Okay, thank you.